Hi, in this video we will be discussing about mechanism of bacterial resistance, and today we will be focusing only on penicillins. Antimicrobial resistance is occurring worldwide and is increasing at a dangerously high rate. Bacterial resistance occurs when bacteria is exposed to drugs. Initially they are sensitive to it, but over time they change and they become resistant. In simple words, bacteria, like human beings, try to survive. So when they're exposed to drugs that are meant to kill them, then over time they undergo mutation so that they become resistant to these drugs. These resistant bacteria start to multiply and form more antibiotic-resistant bacteria. Bacterial resistance may be inherent to organism. For example, gram-negative bacteria are inherently less sensitive to penicillins compared to the gram-positive bacteria. This may be because of their cell wall structure. The gram-negative bacteria has an outer membrane which is absent in the gram-positive bacteria. This outer membrane acts as a barrier to the drug. And also these gram-negative bacteria have a thin peptidoglycan layer compared to the gram-positive bacteria. As penicillins act by inhibiting the cross-linking of peptidoglycan layer, this could be the reason why they are inherently less sensitive to penicillins compared to the gram-positive bacteria. The bacterial resistance can also be acquired either by genetic mutation within that organism or derived from another organism by the acquisition of new genes. Now we come to mechanism of antibiotic resistance. There are three major mechanisms that mediate bacterial resistance to beta-lactams. The first is that bacteria alters the antimicrobial targets. For example, here these are the normal penicillin binding proteins. So when we give penicillin, what happens is that it binds to these penicillin binding proteins and inhibit the transpeptidation reaction, and hence inhibit bacterial cell wall synthesis, causing cell death. And this is how penicillins work. Now, what bacteria does to counter this for its survival is that it alters the structure of these penicillin binding proteins, so beta-lactam antibiotics cannot bind to these penicillin binding proteins. So when it cannot bind, these antibiotics cannot function. However, an important point to note is that this change in structure of penicillin binding proteins does not affect its normal function. That is, these altered penicillin binding proteins can still perform its transpeptidation function and continue its cell wall synthesis. The only thing that changes is that it has low affinity for beta-lactams. For example, these are the mechanism of resistance of enterococci to cephalosporins, meningococci, pneumococci, and streptococci to penicillin, MRSA to beta-lactam antibiotics, and gram-positive bacteria to aztreonam. Now, moving on to the second mechanism of bacterial resistance is that the bacteria reduces permeability to the drugs. So what happens is that when we give antibiotics, they enter the gram-negative bacteria through porins in the outer membrane. So what the bacteria does is that it alters these porin channels. So it can either narrow the porin channel or cause loss or decrease expression of these porin channels, which results in poor penetration of drug into the cell. This makes the bacteria resistant to the drugs. And this is how the gram-negative bacteria, including Pseudomonas aeruginosa, develop resistance to various beta-lactam antibiotics. Now, moving on to the third mechanism of bacterial resistance is that bacteria produces enzyme that inactivates antimicrobials. These enzymes either disrupt the antimicrobial structure or chemically modifies it, and hence interfering the activity of the antibiotic. This is the most common and robust mechanism of resistance for penicillins and cephalosporins. So these enzymes are called beta-lactamases. As the name suggests, they disrupt the beta-lactam ring and hence inactivate penicillin and other members of the beta-lactam group. The enzymes that inactivate penicillins are called penicillinases. Similarly, the enzyme that inactivates cephalosporins are called cephalosporinases. As we know that the structural integrity of beta-lactam ring is very important for the function of beta-lactam antibiotics. So when the beta-lactam ring gets disrupted by these enzymes, the antibiotic can no longer function. So this mechanism was first discovered in Staphylococcus aureus. It was found that the bacteria acquired beta-lactamases and hence became resistant to penicillins. And some examples include resistance of gonococci and staphylococci to penicillin G, Haemophilus influenzae to amoxicillin and other penicillins, similarly gram-negative bacteria to carbapenems, extended spectrum penicillins, and other cephalosporins. And an important point to note is that these enzymes are quite complex and their activity vary against different beta-lactam substrates.